Okay, then hello everyone. I'm Albertini. I'm part of the Imperial iGEM team 2020, and our project's aim is to make lab automation accessible for small-scale labs. Since our team brings together undergraduates from different backgrounds, we encountered the issue of finding a common line of communication to work together. So we were wondering how the software team could communicate and be useful to the wet lab team. We plan this webinar to understand how lab scientists and software engineers have been collaborating both in academia and tech companies. For me, it's a pleasure to welcome here Keltoum and Boukra from LabGenius, and she's dealing with the automation of drug discovery. And yeah, welcome, and please, it's your turn.
Thank you very much. Okay, guys, thank you all for joining today. I hope we're gonna have a good time together talking about lab automation and synthetic biology. I'm gonna share my screen to show you some slides. That's okay. Oh, I actually cannot share my screen. I think you need to give me access to.
Yeah,
Can you enable this for me?
Yeah, you have it.
Oh yeah, awesome. Thank you so much. Perfect. All right. Okay. And here we go. Can everyone see my screen?
Yeah.
Okay. Yeah. Oh, okay, cool. Thank you very much. That's not sure. All right. So yes, first of all, I will just introduce myself so that you know a little bit better who I am before I actually talk about lab automation. My name is Keltoum Boukra, and I come from Paris, and I'm currently the automation team lead at LabGenius, a biotech company based in London, in Bermondsey, more precisely. And I'm also a software engineer here. I have a double background, quite unusual. So first, I have a scientific background. So I studied medicine at Sorbonne University in the biggest hospital in Paris, La Pitié-Salpêtrière, which is historically the biggest health hub in France, basically. And so there I could learn a lot about the body, about the cells, so cellular biology, physiology, biochemistry, but also physics, biophysics. So it was really, really interesting, really, really intense. But you know, the teachers were actually doctors. So they were dealing with patients during the day, and then they would come to us and teach us. And from there, I really had this impression that we could act on the health with a more global approach, and especially nowadays with technologies we have at our disposal. And so that's the reason why I went to robotics engineering. And so I switched to robotics engineering at a Polytech Sorbonne in Paris still. And I could learn about basically how to build robots, which is a mix of many things. It's a mix of mathematics, about computer science, electronics, computer-aided design, mechanics. So it's very multidisciplinary and it's very interesting. And so knowing about my background, you probably understand a little bit better how I ended up at LabGenius, which is also very multidisciplinary. So here, what we do is really a mix of 3 different areas, which originally don't have much to do together. So it's machine learning, synthetic biology, and robotics automation. The ambition of the automation team here at LabGenius is really to turn science into production, make science as predictable as running a code on a computer, being able to control fully the outcomes of an experiment. And the idea of it all is really to bring the human outside of the execution loop. We don't want a human to be here to repetitively do the same tasks again and again. We really want here at LabGenius that the scientists really focus on developing new assays, on basically using their brains to do things that we need to create. And then we want the robots to just execute these things. And so I thought maybe we could start with an overview of what is the traditional drug discovery process, because that will help us to understand better where LabGenius is trying to focus. So if you look at the drug discovery process, it basically looks like a funnel. So you have different stages, and you start with many compounds, and in the end, you hope that you will find the one drug that will be effective on a disease. But the problem with this process is, first of all, the time it takes to find this new drug. It takes roughly between 10 to 20 years to get from the stage where we decide to create a new drug for a certain disease to actually be able to give this drug to patients and sell this drug on the market. The second really important thing about the drug discovery process that needs to be improved is the cost. Depending on the studies you will read, the cost is slightly different, but one of the latest estimations is that the cost to make a new drug from scratch is about $2.8 billion, which is huge, right? And finally, what are the chances of success? Well, even if you spend time trying to develop a new drug, even if you spend money trying to develop a new drug, the likelihood of getting to the point where you can give this drug to patients is still low. Typically, between the second and the third stage of the drug discovery process, you have only 10% of the candidates that actually make it to the next stage. So the likelihood of failing is very high at that stage. And so if you look a little bit closer to this process, you have four main stages. So the first one is the research and development. So there you identify the disease, you know which disease you want to target. You also need to find out what is going to be the target in the body. You need to start screening for compounds. So you will make many different combinations of depending on what type of molecule you're working on. Let's say with proteins, you will make many different variants of proteins, and you will screen them. So you will try to find which ones are the more likely to become drugs. And that leads us to the next stage, because only a handful of candidates from these 10,000 compounds or more will actually proceed to the next stage. And the next stage is the preclinical study stage. Here, we are going to test further these best candidates from the previous stage. And this happens in two ways. First, in vitro, and then in vivo. The idea in both is that we want to understand better the characteristics of these candidates. We want to know, for instance, what is their efficacy, how quickly they're going to be absorbed in the body, but also eliminated from the body, how toxic potentially they are for the body. Once we identified again from this stage a couple of good candidates, we go to the next stage, which is the clinical trials. And now it becomes more serious because it involves humans. And there are three phases. Each phase will include more and more people. The idea here is that we are at that stage with about 5 candidates, maybe less. And hopefully, if everything goes well, after a couple of years, we will be able to ask for an approval. So typically in the US, it would be the FDA. And yet, once this is done, once the drug is on the market, there's an indefinite period where we're going to monitor the long-term effect of this drug. So you understand now a little bit better how tedious this process is, how long it is, how expensive it is. And here, there's an important aspect which really helps. It really helps to have it in mind when you're working in this area. It's the developability of the new candidate. As early as the process, you need to find out how manufacturable this potential candidate is. Are we going to be able to find a candidate that is really good, but that is going to be breaking down as soon as we make it appeal, or a different appearance? How specific the binding is as well. So I said earlier that we'll have to find a target for a specific disease, but we need our candidates to bind very tightly to our targets, and we cannot afford to have our candidate binding to other sites in the body, right? And so this is really important, because having non-specific binding might be very bad for the body, right? It might actually have side effects that are obviously not desired. And so the earlier you find out about these factors, the better it is, because as I said earlier, that the later you find out these things, you would have spent so much money that it's not worth it. So the earlier you find out about the developability of your candidate, the better in terms of cost. Okay, so how does LabGenius approach this? First of all, we really focus on the first stage and slightly on the second stage of the drug discovery process. And we build this engine that we called EVA, which is, you probably know about the synthetic biology cycle, so the design, build, test, learn cycle. This is basically what we do. We have a platform that has basically 4 stages. The first one being the one where we're going to design sequences. The second one where we're going to go in the lab and build them. The third one where we're going to test these candidates. And then the last one, which is involving machine learning, and where we're going to try to understand what happened in the previous cycle and predict what's going to happen in the next one. And so if you focus, for instance, on the proteins, so proteins can be drugs. And there are 20 amino acids that can form a protein. The question here is, how many combinations are they? If you have to combine 2 amino acids to make a protein, it's 20 to the 20th. So that's 400 possibilities of different combinations to form your protein. If you have 3 amino acids to form your protein, you reach 8,000 possibilities. And the more you go in terms of number of amino acids, the more possible combinations there are. And knowing that typically most of proteins are longer than 100 amino acids, that becomes a huge number of possibilities to design a new protein that would potentially become a new drug. So this is why we needed to think, when we build the platform, how to be able to screen as many candidates as possible, and in the best way as possible. And so this is why we combine both automation and also machine learning. And so, as I was saying, this is really what we do. First, we design new libraries. We use our computers with different strategies, and we'll come up with thousands of sequences that we think are potentially good candidates. We'll go in the lab, build these candidates and form libraries of candidates, and then test them. We will use our robots to test these candidates in order to screen these candidates in order to understand how well they perform. And finally, we'll retrieve the sequences of these candidates using next generation sequencing. And so we do that again and again, until we really improve the performances of the candidates we are working with. And finally, in the end, we'll come back with brand new compounds which we really hope are going to be future drugs. And so, as you understand it, it's really a multidisciplinary work that's happening here. Combination of synthetic biology, machine learning, robotics, all working together. But where does automation come into play? This is what I would like to talk about from now on. How do we use automation here? And so first, let's think about the reproducibility. So here are some key numbers about the reproducibility in science and in the drug discovery process. So there are 2 surveys. The first one is about, so it's 1,576 researchers have been asked about their research. And so 70% of these people answered that they failed to reproduce someone else's experiment. And half of them also answered that they were not even able to reproduce their own work. And so another study showed that the amount of money spent on non-reproducibility preclinical research is estimated to $28 billion in the US alone. So this really made us think about reproducibility and really gave us confidence that we should invest in automation when building our platform. And so, as I said earlier, the vision really is to make science predictable and manufacturable. We want to have a couple of inputs, a couple of outputs, and robots in between executing protocols. why do we want to use automation? These are four, let's say, benefits to using automation, in our opinion. The first one being high throughput and repeatability. Sorry, we're able to run processes where we can process 96 or 384 candidates at a time. And that will take the same time as one scientist doing it by hand for one candidate sometimes. So being able to run many more candidates at a time, screen many more candidates at a time. Save time, obviously, because we don't have to stand by the robot for the whole process. We can set up the robot and just walk away. We don't have to spend more time running experiments, running processes that have been standardized and automated. Reliability and precision, because obviously when you work in this kind of, when you do this kind of work, you want to be able to be 100% sure that candidate A has been treated the exact same way as candidate B. So you need to apply the exact right amount of protease or of cells or whatever it is, depending on your assay. But precision is really, really key. And finally, cost savings. And this is linked to the fact that when you have automation in place, once you pet for these robots, there's no human involved. So in terms of, just if you think about it as in terms of people, it saves costs. And so I said earlier, we are a platform that LabGenius has a platform that uses machine learning. And anyone who works with machine learning knows that data is really necessary, is key. Without data, there's no machine learning. And this is one important thing as well that I would like to highlight, linked to the automation. When we use robots, we're able to track much better what's happening. We have log files of exactly all the steps that happened on the robot at what time, and this is really, really helpful. This, you don't have access to this when you perform work manually. You will potentially try to remember, take notes, take pictures, but it will never be as good as getting log files from liquid handlers or various machines we use. And so this is an overview of the different devices we have here at LabGenius. Some of them are here in duplicate. We have many, for instance, of this Felix from Analytica. So we have a mix of liquid handlers. We also have several robotic arms. We have all the machines that you probably know about, so incubators, PCR machines, qPCR machines. We have all of these machines, but they are automation friendly, which means that we can use them in combination with other machines, thanks to the robotic arm that will link them together. And so sometimes it doesn't work really well. Sometimes it's awful. You come back to the robot and you don't understand what really happened. And these are tests I will mention. But most of the time it looks pretty good. So this is like an overview of the different things we usually do on our platform. So you see on the top right corner, this is the 3D printing station that we have. So on top of the robots I showed, we also like to use 3D printing for different things. Automation is obviously becoming quite you see automation in place quite often now in the biotech companies, but still it's quite often the case that we need to build our own devices in order to do things on the robots. Typically some plate holders or very bespoke designs that we need to then print. If you look at the bottom, you see the robotic arm in action. So here we are running an assay which consists in quantifying the proteins. And so the more purple color you see, the higher the concentration of proteins is. So yeah, this is really to give you an overview of the different kind of things you can see when you work here at LabGenius in terms of automation. But I would like to finish with a very specific example. So this is an example of a pipeline that we've automated. So as I said, we make use of next generation sequencing, which involves some work before we can actually go to the sequencers and start the run. And so the question we're trying to answer here is, how can we feed our machine learning algorithms with data? And the data typically come from the sequencers. And this is typically, not only, but typically at that stage, the test and learn stage of the cycle. And so this is how it looks. Okay, so I said we design libraries of candidates, right? We start there. And then we apply different pressures on these libraries of candidates. So we're talking here about millions of different potential candidates, right? Millions of individual variants. We'll apply pressures using a Kingfisher, which is an automated machine as well. And out of these selections, we will want to find out what are the sequences of these best performers, the best candidates, the ones that survived all these pressures. And so this is where next generation sequencing comes into play. We prepare these libraries of candidates that survive to the selections, and then go to the sequencers and sequence these libraries. And so how does it work? If we go a little bit more into the details, first stage is really the amplification. We have some libraries of candidates before and after selection. So the first one being really big, and the next one being smaller. And we'll try to amplify every single variant in order to be able to then sequence. And so we use a two-step PCR for this. And so you probably are very familiar with the PCR method, but I will just go through this very quickly. So PCR is really a mix of some DNA, so the DNA you want to amplify, some primers, which are shorter DNA sequences, a couple of nucleotides and enzymes to make the reaction happen. And all these go in a well or in a tube. And then we'll go through a couple of cycles of polymerase chain reaction, so PCR, where we'll apply 3 different temperatures, and this many, many times. And the second step after having amplified the sequences is to purify them. So obviously, because as I just said, we have a mix of different things in this tube or well, we want to get rid of all the undesired sequences of DNA or nucleotides. And so we'll use specific methods based on magnetic beads, where the beads are going to attach to our sequences of interest, and we're going to remove all the rest. And so typically, how does it work? We let the beads incubate with the mix post-PCR, and then we'll place the plate or the tube on a magnet. And so the beads will be attracted by the magnets. And so then we'll come in and remove all the, what we call supernatant, what is not attached to the beads. And then we'll wash a couple of times, typically with the ethanol. And finally, we will go on to the elute stage, which is to now separate our DNA of interest from the beads. And finally, we obtain something really clean, which is what we want. And so this is how it looks on the deck. So this is a typical example of use of the robots. So here we pick only 8 tips, but we have different capabilities. We can pick up to 96 tips. And so here is the stage where we'll perform the washes. So we mix the beads with a wash buffer containing ethanol. And so we'll mix many, many times. And then the next step, as I mentioned, is to separate the beads from the rest, from the dirty stuff, basically. And so for that, we use our on-deck gripper. So here we grab the gripper, the head goes down, take the gripper, and then go to the plate, grabs the plate, and remove it from the magnet. This is very similar to what you would do by hand, but the difference is obviously that it's automated, and also the number of libraries that you see. So here it's 96 that you can do at a time, compared to by hand, which is typically in tubes and therefore much longer. And finally, the final stage of the NGS sequencing pipeline is the quantification. And this is a really, really important point. In order to maximize the quality of the sequencing and also maximize the use of the cartridges we buy for the sequencers, we need to be able to quantify very accurately how much DNA we have. And so for that, we use a qPCR, so quantitative PCR. And typically, when you set up the reaction, you then go to the qPCR machine and get a similar curve to this. So you would have an exponential curve for each of the samples. You would have no signal from the nota plate samples. And for each of the samples, we want to look at the CT value, which is the beginning of the curve, the exponential curve when it goes up. And so how does it look in practice? Well, it looks like this. So we have a couple of plates. We have some adapters on the deck. And this is what happens. So first step is to dispense master mix containing different enzymes on the plates. So we'll distribute this master mix across the plates. So we'll fill up the 96 wells with this master mix. Well, then we'll perform a couple of dilutions of our input libraries and then transfer them into these reaction plates. And finally, we go to the PCR machine and then get this list of CT values that I was just mentioning. One key thing about automation and lab automation is that you need to be able to deal with the data. So yes, it's great if you can be able to increase the throughput from 1 plate to 100 plates, but then you need to be able to analyze the data. And so this is something that we're really focusing on. We want to be able to analyze whatever comes out of the machines when we perform automated runs. Now that we've quantified and we also amplified our sequences, we're able to sequence our sequences. And so for that, it's very simple. We'll take a mix of our prepared samples and load them into the cartridge. If you remember the slides I showed earlier about the benefits of automation, well, here it's very true in this example. So if you look at the throughput, for instance, before we automated this pipeline, the throughput of the sequences were really low or much lower. And now, because we increased the capacity of the team to prepare samples for sequencing, we bought another sequencer, and we are about to buy another one. So this is a typical example of, using automation for higher throughput. Time saving, because obviously, again, you set up the deck, it takes you between 10 minutes to half an hour, and then you're basically done. You walk away, you don't have to stay around. You can do something else. You can set up another robot or work on developing a new assay. Reliability and precision, which is absolutely key in this protocol, as I explained. You don't want to overload your cartridge, because that would be a disaster in terms of quality for the sequencing. And so the robots do that really well to distribute the master mix, for instance, for the qPCR in a very reliable way, and then dilute the different candidates in the right with the right ratio very accurately is really key. And finally, cost savings, as I said earlier, in terms of human, but also if you think about it as, if we are not able to prepare these libraries the right way, we'll not make the better use of our cartridges. And these cartridges for next generation sequences are extremely expensive, right? Also, the cost of having to redo things when the results don't look as they should look. When we are running these processes, we are 100% confident. We know what's going to happen. We know that the standards are going to look exactly the same as the previous run. And so that allows us to not have to repeat things, which is also a cost saving in the end. All right, so I hope you enjoyed it, and I will be happy to answer any question. And just note that we are hiring actively, including in the automation team. So just drop us an email if you're interested. Thank you very much for your time.
Thank you. Does anyone have any question? Feel free to unmute yourself and.
Hiya, a really great presentation. Thank you so much. Someone's just asked in the chat, but I'll just finish. Have you been able to make it any way through that research preclinical trial, clinical trial, etc. process? Or if you're allowed to disclose that, have you had like tangible kind of, yeah, it's so much faster or anything like that?
That's an excellent question, really interesting question. So it depends on what we are working on. So here at LabGenius, there's a couple of different things we do for each project. So some projects are done in collaboration with pharmaceutical companies, and there we'll agree with them on, okay, how far do we push the discovery? When do they take over, if they take over? And another side of it is, another kind of project is when we develop new proteins or new candidates internally. And so then we decide when we want to stop pushing and when we want to find a partner. If I can give you a precise example, so there's one project that we're working on that we are now going to push into the next stage, so preclinical work, in vivo work, and so this should happen in about a quarter, hopefully. So there we will be able to work with actual animals to see how well we are able to predict a candidate.
Thank you.
Yeah, Jay is asking how much do the machines cost?
That's a good question, because I know in academic it's often hard. People, when they first join LabGenius, they come in the lab and they can't believe what they see, which is quite nice. I've never been in academic before, so I don't really know. We have quite a wide range of costs in terms of machines. So one of the robots that I showed in the slide, OpenShrone's OT1 and OT2, they cost about £2,000, which is really, really cheap. But you can go as high as you want, I think, in terms of cost. You can go, there are robots that are about £200,000 that are very, very used, I think, in the industry as well. So yeah, in average, I would say around £30,000 for a good liquid handler, and then depending on which machine you're talking about, so plate reader, robotic arm, different costs. But if you're an academic and you're looking for a liquid handler, OT is probably a good one, so OpenShrones.
Any other questions?
Hello, thank you very much for the talk, very interesting. I had a quick question when it comes to the development stages. From what I understand, you concentrate mostly on the first two stages. What sort of techniques do you use, for example, in machine learning to improve outcomes in these stages?
Yeah, that's a really good question as well. So how do we use machine learning? It really depends on the project and the stage of the project, right? So at the beginning of a project, machine learning is basically linear regression. We are trying to understand the outputs of sequencing in a very simple manner. But the more we iterate over the cycle that I showed, the more we're able to build models that are really complex, and also the better these models perform. And the techniques we use are really dependent on the project we are working on.
Thanks.
There is a question from Zoltan asking, how would you compare LabGenius with Ginkgo Bioworks?
I must say I don't know much about Ginkgo Bioworks, so I'm not sure how to answer the question, sorry.
Okay, great then. If Anyone have any more question? Or we can continue with our webinar and our program. Okay. Okay. Now the schedule will continue with Mike Fero, who is the CEO of TeselaGen, and he has both experience in physics, with studying elementary particles at CERN and at SLAC, and also in synthetic biology. So welcome to the talk, and I'm curious. We are all curious about your story.
Thanks. Can you hear me okay? Does it sound all right?
Perfect.
Great. Well, I guess I should try to share my screen, right? Let's see if we can get this to work.
Yeah, Keltoum, can you make him host, please?
Yes, I will try to do that. Sorry, I didn't anticipate this. How can you try now? Is it okay?
Sure, let me give it a shot. Share screen. Okay, I'm just going to share my whole desktop. Apologies if there's any weird stuff on it. Let's see. Now I'll see if I can. We'll know it works if you can see a black and white. Is that coming through?
It's good.
Is it working?
Yeah, that looks great.
Okay, cool. So yeah, thank you very much for inviting me to give this talk. It's really a privilege and a lot of fun. And I really liked hearing what the last speaker said because it saves me a lot of time. That was a very good introduction to the idea of a new way of doing drug development. For us, it goes a little bit beyond drug development. We try to apply our technology to industrial biotechnology as well. So folks who are producing, say, high-value enzymes or chemicals, things like that. But it's really the same idea. And I guess in the spirit of the last speaker, I'll tell you just a little bit about myself. So yes, I was a physicist up until I still am, I guess. You don't actually lose your physics credential when you switch to biology, but I switched focus in the late 1990s from particle physics at CERN and SLAC to biotechnology. And it's complicated. We can all get past COVID and go have a beer someday. I'll tell you about all the crazy decisions I had to make to do that. But the upshot was that I wound up at both biotechnology companies and also back at Stanford University working with David Botstein, who was the chair of the Department of Genetics at the time, as well as Pat Brown, who was developing something called a gene chip or a gene microarray. So I helped Pat and David develop some of the first gene microarrays in the late 90s and early 2000s. Pat has since left Stanford and went on to found Impossible Foods. I left Stanford about the same time and founded TeselaGen, which has done fine for the last 10 years, but we're certainly not Impossible Foods with the Impossible Burger and all that stuff. We focus almost strictly on software, so we don't produce a molecule, we don't produce a product. So that gives us the freedom to go into companies and really help them out in a very intimate way to scale up their operations and save tons of money. So a lot of these companies, when they go, synthetic biology companies, they'll burn like 10 to 50 to $100 million sometimes in cash, just setting up and executing on on advanced informatics. And we can save them a huge amount of money by helping them develop that. So when we go into a company, let's see if I can switch slides successfully here. This doesn't seem to be toggling. There we go. So yeah, so when we go into a company, we usually see a very heterogeneous state of affairs, which is typical of an R&D, the R&D side of a commercial operation. You know, you have data scientists and automation and scale-up facilities and data science and all that kind of stuff. And what we try to do is put that into a bit of order. So as the last speaker alluded to, it can take quite a while just to do basic R&D cycles. When you're developing a molecule, you know, you have R&D team, you have human technicians, analytic instruments, you have liquid handling and so forth. Typically, you're not handling that much data. Some of the things that we enable is people to handle much larger amounts of data. So we kind of clean up and straighten out and apply our software so people can do this much more efficiently. And our platform is based on something called iterative machine learning. So it's a technique that's been used in other scientific endeavors more than it has in biology, quite frankly. So we're sort of one of the companies that's bringing this to biology, and we have actual recent paper in Nature Communications that came out a couple of weeks ago about a publication we were able to do with the Danish Technical University and Berkeley Lab on how you apply this to molecule development. And what it amounts to is acknowledging that you can make a lot of progress in biology by exploiting some of the, you know, the beautiful benefits of biology, that you can do high throughput screens and you can do genetic selections and you can search really vast hypothesis spaces for the candidates that are going to be your successful molecule. As you heard in the last talk, there's often a pipeline approach. At a certain stage, you push a certain candidate through. You can take slightly more holistic approaches when you use this kind of sort of more high throughput hypothesis-based search approach. You don't necessarily have to throw anything out too early. You can learn from things that fail, actually, even fairly far down the pipeline, and go back and based on the model, pick other candidates or search other possibilities for something that might work better. So yeah, so it's often the job of the company we're working with or their scientists to define the parameters of the hypothesis space, since they are the domain experts. But then we can help them with this process of actually building models as soon as possible, and then training and then predicting where to look next. If that makes sense. Yeah, we've been doing this for a while, so we've developed some nice machine learning techniques to try to optimize these molecules and try to search these spaces. We use a lot of sort of modern techniques that a lot of the basic techniques have been around since I was doing machine learning back in the 90s. But what we have in the 2000s are sort of another layer of tooling that helps you use those machine learning techniques much more efficiently. So it's been kind of nice to see the sort of layer of tooling that's been built. A lot of it coming from, sadly, you know, people trying to do social media engineering and facial recognition and stuff like that. But you know, a lot of it's hopefully useful for making the world a better place too. So the platform we built is a 4-part platform, has a design module, a build module, a test module, and a machine learning module. And a lot of people ask us, well, you know, why did you bother going and building all of this infrastructure when what you really are focused on is, you know, machine learning and molecule optimization? And my answer is fairly straightforward. If you really want to do machine learning right, you really want to control the design of experiments. So you can control statistical error, and you can really predict whether you can even do a machine learning exercise or not. And it captures basically your independent variables in a very you know, accurate way. So you define your sort of genotypic space, if you will. And then by controlling or monitoring the build. So unlike the last speaker, we don't actually, you know, we don't actually have robots and stuff like that, but we tell the robots what to do, and we gather data based on how they're doing back into the system. And the reason for that is for us to control systematic error, the other large class of error that we want to control in a machine learning experiment. And we have a real advantage here, folks. I mean, as opposed to folks who are doing, say, genomics and are just having to take like epidemiological data or data from various people's genomes or whatever and try to gain insights from it. we're doing a forward designed experiment. So we have control over the design of experiments. We know what the statistics are. We can monitor the systematic errors. So that's the big advantage that we have as people on the R&D side. And then the test module then is for us, we call it the test module. It's really just a big data lake where we transform the data into a format that is extremely easy for machine learning people to use. They don't have to like do a lot of what we call data engineering, which is not the same as the science part of it. It's mostly just wrangling, you know? So that's what the test module serves as, that place where we can gather that data in a way that we can apply our models to it directly. And then finally, the evolve module is where we actually execute our trained models and give people new targets. And this is all software that's been built, and I can even show you, if people are interested, a quick video of someone doing a molecule optimization exercise through the system. And another important thing to note is that that we built the system in a modular way, so you can take advantage of different parts depending on what it is you need. And we also have built it with an eye towards integration. So we have really great APIs and other techniques which are more technical for integrating our solution with customers that may be using LabGenius or maybe using Benchling or other tools to do various aspects of what goes on in the lab. Yeah, this is just a little bit on the architecture. We have our own machine learning architecture up in Google Cloud. Well, it's too much, but it really gives people a large amount of power for a much cheaper price than it, you don't have to spend Ginkgo or Zymergen type money to, you don't need to spend $100, $200 million to get this type of infrastructure anymore. And it's just more technical stuff. I probably should have taken the slide out, but just shows how, we queue stuff from the various modules into the workers in our cloud-based system. And I guess I've already alluded to this. I'll go into a little more detail. A lot of people talk about APIs, application program interface. We have several other layers of ways to integrate with our system. We have something called a CLI tool. API stands for application programming interface. So it's a way that a computer can talk to another computer through a program. CLI stands for command line interface. You can actually go right to the command line and issue commands to our system and get responses. And that's great for people who want to do scripting. And that helps with, say, handoffs to LabGenius or automation or different things like that. We also have something called a microservice framework, which is a way that we can, a type of tooling that we can use to wrap your proprietary software. So I know you're probably academic at this point, but when we go into companies, they often have some cool stuff they develop and they're kind of secretive about it because they really worked hard on it. And they want it to run behind their firewall. So we can wrap that in a microservice worker and it can run behind their firewall all nice and secure. And it looks like it's running in our platform, even though our platform's running, in Google Cloud on some server somewhere. So that's kind of cool. So I can spend just a little bit of time talking about some of the modeling exercises we've done. So we have this notion of predictive modeling. So you've defined a fairly large hypothesis space and you start to build a model that relates genotype to phenotype. And then we can use that to recommend to people where they go next in that space to find another possible candidate. And this really helps with people who are doing optimization like titer prediction and optimization, because you're not really going back all the way to square one. You don't have to go back to design. You really have designed the hypothesis space. You just want to search that space really efficiently. And this is actually something that's led to a nice publication with, as I mentioned, the Danish Technical University, as well as Berkeley Lab. And that just came out a couple weeks ago. I guess if we're going to get a recording, if you get a recording of this, you'll be able to grab the reference and check it out. This was really nice because a lot of the work we do, which I can tell you a little bit about with commercial companies, it's proprietary. They don't really talk about it. But this was an academic exercise for someone who was doing their PhD at DTU, and they did a secondment, I guess. at the Danish universities, you have secondments, and you actually go to a company for a few months and do time there, which is great. It's a great system, and they get their PhDs out quickly too, which is also great, as opposed to the American system where you can be doing your PhD forever. So it was really awesome. So it led to this paper, and we were super stoked about it. Yeah, so this is just some screenshots from the paper. We basically were trying to use a combination of predictive and machine learning techniques. So there's, excuse me, a combination of mechanistic and machine learning techniques. So there's some parts of it are like, okay, we know basically what's going on in yeast metabolism. This is the space where we want to learn, or we want to search, right? And then in that space, we created a certain number of candidates that we were going to search to see how we would optimize the system, and from that predict where we should go next. Because this was an academic exercise, we didn't have a lot of money. It's basically one loop through the DBTL cycle, you know, prediction, and then we were able to test the prediction. Ideally, what you'd like to do, and what we do at companies, is you go through several iterations of this. But for the purpose of the publication, we did basically one loop. Yeah, that's just, this is from the paper as well, so nothing really new there. But I would like to talk a little bit about, you know, this is fine, sounds very academic, and yeah, it's great to get more tryptophan out of yeast, but there are good reasons to, in industrial biotechnology, to try to optimize this type of technology. And I'll just go into a little bit of an example with another company that we work as a cool system for removing greenhouse gases. So I'll just it's a little bit of a tangent, but I think it's kind of fun to talk about. So as a species, we consume a hell of a lot of oil every day for transportation. And as a result, we, you know, emit something like 32.5 gigatons of extra CO2 into the atmosphere every year. And as much as coronavirus has kind of consumed us for the last year or so, or 9 months or so, this is still one of the big problems. And we're seeing like the sort of, it's not sort of early warnings anymore. We're actually getting real effects. The fires in Australia a couple of years ago, the crazy fires that we've had in California this year are sort of, you know, telltale signs that the climate is changing, and we should, you know, really pay more attention to what's going on and help remediate this problem. So those numbers are kind of abstract. You can sort of estimate that a ton of CO2 would completely fill your house with CO2. That's kind of what a ton of CO2 is. And 32.5 gigatons would completely cover a small state, about a mile under a blanket of CO2, a mile deep. So it's a lot of extra gas. It really is. So steel production counts for about 5 to 7% of global greenhouse gases, and China makes about half of the world's steel. So we work with a company called LanzaTech out of Illinois. They originally came out of New Zealand, but then they moved to the States, and they have an extremophile that'll eat carbon monoxide and make ethanol. So instead of flaring this flammable gas into the air and making CO2, you basically get to take another crack at it. So it's kind of like a use, reuse, recycle type of thing. And it actually takes a lot of cars off, the equivalent of a lot of cars off the road if you do this. So we've been helping them with their microbes, optimizing them, and also trying to make even higher value products. Well, ethanol is not that high value. Try to make high value products using this microbe. So they have I guess I mentioned this, they have a gas sort of reception tank. They compress it, they just bubble it through this giant fermenter which has, you know, Clostridium species growing in it, and they make ethanol. It's kind of crazy. When I was there at LanzaTech, I saw the sort of inoculum, the thing that they use to inject the bacteria into the fermentation unit, and it's like a PCV pipe that's probably 6 inches in diameter and 3 feet long, like this giant bullet, and with a handle on it, and they like shove the thing in and turn the handle and inject all these bacteria into this thing, and off it goes. It's really crazy. So yeah, we've been working with them for several years now to optimize their, and this kind of goes into this sort of large category that's not biopharma, as we heard in the last talk, but it's more about industrial biotechnology, using microbes as, itty bitty factories to take, some carbon source, whether it be sugar or a greenhouse gas, and turn it into some useful product. For us, the greenhouse gas thing is extremely interesting because it obviously has, beneficial effects in terms of climate remediation. And it also, from a business point of view, it's usually a very cheap feedstock. The cheaper your feedstock, the more chance you have of making a commodity product, feasible in the marketplace. Okay, so I have another case study, which is discovery of novel AMPs. These are antimicrobial peptides. We heard a lot about the biopharma sort of pipeline. I won't go too much detail about this, except to say that we work also with biopharma folks who are doing optimizing molecules, which have a therapeutic effect. So we've done a lot of work around AMPs with our customers, assessing the fairly large libraries, and then trying to hone in on the ones that are going to be the candidates to take forward. Yeah, so I think we'll just kind of slide through this, since we heard a lot about biopharma in the last talk. We also heard about antibody optimization, so we can also use our technology for that. We can start with a single antibody candidate or a library, start to create models for that, and then suggest new places to look in that hypothesis space. Yeah, so I have a couple slides here which you guys can check out. I can make a PDF of this talk available to the organizers, so you guys can check this out. Let's see, protein expression prediction, another case study. Oops, I think that was the end of my talk. Sorry, I just put this together this morning. I don't know, I think I just hit a key and it hopped out by itself. Let me just go back in. All right, so yeah, I won't go through every single case study, but I'll just say that, just bring you guys up to date at where we're at TeselaGen. As students, you're probably interested in knowing where we're at as in terms of development of the company and whether we're hiring and stuff. So our design module is where we started. That was the technology that we brought out of the Kiesling lab at Berkeley, which was automated protocol development. So our design, so we can take a sort of a design of experiments, turn it into a digital protocol, and then send that directly to, automation. Once we had that, we had a Fortune 50 company that said, hey, they didn't like the existing LIMS systems. So that was the genesis of the build module. We took the designs that were automatically generating and converted them into machine instructions. And then we wound up building an entire LIMS system because, once we'd done the hookup to automation, the rest of it was, was not that hard. And then the test and evolve modules, these are kind of a little bit more in stealth mode. We use these with, important clients where we're developing molecules. So I would call, in terms of unleashing them on the general public, I would say that they're sort of advanced beta, but we do use them all the time with, with our customers, our commercial customers. So yeah, so that's kind of where we're at in terms of developing the platform. And let's see, this is the last slide. Is a nice pretty picture of San Francisco. We're based in San Francisco. We hire mostly out of Stanford and Berkeley and the local UCs because the cost of living is so high in San Francisco. A lot of folks, we hire already there. But that being said, we have people actually scattered a bit all over the world. So we can, and coronavirus has really made it quite obvious that we can work remote. So we have people who work with us in Europe and in this Midwestern part of the United States and the East Coast, as well as South America. So yeah, we're pretty virtual company in that sense, at least for the last 9 months. We all went home and stayed. Yeah, so that's it for TeselaGen. If there are any questions, I'm happy to answer or go into more detail. I can show a little video if people want to see that. I'll just leave it up to you guys.
Yeah, please unmute yourself if you have any question, or writing in the chat.
Hi, it's Keltoum again. Thank you very much for the talk. It was really interesting. And I maybe don't have a question, or maybe I have a question. But first, I would like to say that it's really impressive to see how able you are to use one single platform and apply the platform or the use of the platform to different domains. I find it really, really interesting and impressive. Maybe one question would be, you mentioned several times automation, but I'm just trying to understand how do you link your work to automation? I mean, where does automation come into play in your platform? Are you a software company, or are you a combination of software and automation?
We're strictly a software company. So I don't know if am I still sharing my screen?
No, you are
I'm not sharing.
Let me share it again and just show you what I mean by that. And maybe this will help you see where the handoff is. So can you see my screen? Is there should be like a Vimeo should be up right now.
Yes.
Yeah.
Okay, let's see if I make it full screen, can you still see it?
It's looking good. Okay,
cool. All right, so I'll scrub through this real fast. I don't want to bore people with it. You know, this thing goes on for like 10 minutes or something, but this is the design stage, design module, which was our original software, was for creating sort of a design of experiments of a large hypothesis space of possible genetic modifications. Metabolic pathway modifications, that you also domain modifications or whatever. And then what this leads to, let's just jump ahead now, scrub. This is picking the various genetic elements. Okay, right here where it says, let's export. So what we do is we export a digital protocol to our build module. And the build module can then pick up that protocol and run with it. And where we hand off to automation is in the generation of a work list. So this is also showing a kind of a cute thing where you can go to Twist and other DNA vendors and check their buildability index. So a lot of these vendors can't build this stuff, and you have to go back and redesign it. And so there's a way of doing it really fast. So you don't have to spend days and weeks trying to find out if a vendor can build something for you. So anyway, you check and make sure that they can build it, comes back with no errors, then you continue with your workflow. And then there's a specific point in here. Let me see if we can find it. This is a lot of just Lindsay stuff, ordering DNA, getting it back into your system, running your workflow. Let's see. Action planning, plate wrangling. Keep going. Okay, work list generation. So this is the key. So this is the part where work list that's tuned to the particular robot that you're using gets generated as a CSV file. We can also do integrations through CLI directly to the robot, but whatever. The most important thing is that you get this file. Once you have this file, you're off to the races with your robot. And so we've done some really nice integrations with, say, the Echo, which is a liquid handler, which is really tiny, picoliter amounts of liquid. So that's one that people really like to integrate with. Yeah, so that's the handoff. I hope that answers the question.
Yeah, that's great. Thank you very much.
If I scrub forward a little more, you can see that a lot of this is kind of boring. It's just doing a lot of Lindsay stuff. But if you scrub forward to the end, you see that what you finally come out with is a plate of constructs. And those are the constructs that you then assay. And then the assay data goes into this next module, which is the test module, which holds that data for you, right? The test module isn't doing anything with the data yet, except holding it and making sure it's formatted correctly for your machine learning team. And then the machine learning team can pick it up. We have integrations with Jupyter Notebooks, if you're into Jupyter Notebooks, and then run with it. And then we deploy the model through the Evolve module. So and then I'll just scrub ahead again to the Evolve module. And so this is the Evolve module, which actually, so the idea is that the biologists don't really care about your machine learning problems and how much effort you had to put into it and all the model building you did. They just want to benefit from it. So what we do is just deploy the model in the Evolve module so people can, run their data through it and get a prediction. If that makes sense. So any more questions about that kind of workflow, what we're trying to do?
Hello, I do have a quick question about the data format you use in your software.
Yeah.
And in particular, if you are aware of SBOL as a standard in synthetic biology and what you think of the various standards currently used.
Yeah, we actually use SBOL. We were one of the early adopters of SBOL. So we've been around, we're kind of one of the oldest synthetic biology companies. Not the biggest by any means, but yeah, we're on the SBOL industrial committee and we follow what they do. Sometimes we have to go a little faster than is often a fairly academic exercise, and we have, stuff we have to get done really quickly. But the nice thing about SBOL is it comes along a little slower, but it kind of creates a little bit more organized atmosphere for the industry to operate in and more standardized type of stuff. And then we kind of slowly convert over to SBOL way of doing things over time.
Thank you very much.
There is a question from Zoltan who is asking you that you've mentioned a digital protocol. Is it a proprietary format or something open source?
It's open source. I mean, people usually don't, because it's just integrated with build, people usually it's just a JSON file, so there's nothing really proprietary about it. But usually people are using build too, and they don't have to worry about it. They just yank it over from design.
Okay. Thank you very much. It was a pleasure having you here. And now we're gonna continue and try to have a look what's going on in academia instead. And our next speaker is Michael Crone from Freemont lab at Imperial, and he's going to share his experience developing a PCR platform for COVID testing, optimizing the workflow. I don't want to say anything more. And, thanks, Michael.
Thanks very much. Okay, let me really.
Can you?
Yep. Cool. Okay, so maybe this is slightly different because it's not just about the software development. It's also about kind of what are the considerations if you're trying to develop a test for COVID, I suppose, in a pandemic. So something that needs to be really robust and reliable. So just briefly about briefly about myself. So I actually have a medical background. So full doctor already, but then I did a master's in systems and synthetic biology at Imperial, and I'm currently doing my PhD. And I work at the White City campus in the London Biofoundry. So we have lots of different automation equipment. So acoustic liquid handlers like the Echo, as well as normal liquid handlers such as the synbio Felix. We've got other so we have plate readers integrated into these platforms. So you can use these robotic arms to move plates around and things like that, schedule experiments. And so essentially what I'd like to talk about today is how do you kind of bridge the gap between academia and the National Health Service? So trying to design a reagent agnostic automated diagnostic workflow for COVID testing in a pandemic. And so we started working on this in January, but just probably February, March, you started to see these kinds of headlines that there were massive problems in terms of shortage of reagents and lots of people kind of taking advantage of the situation. And so we just thought we'd like to help out. And the problem is a lot of these diagnostic labs are kind of very bought into specific platforms. So at least the Imperial NHS Trust, who we have done work with, have Roche, Abbott, and Oz Diagnostics as their main platforms. But for example, Roche only has enough capacity, well, they supply 36,000 or something tests to the UK every week. Abbott also has limited supplies, so you can't solely rely on these normal kind of your diagnostic workhorses, as it were. And we also, like, we are at Imperial College, but that is very, very different to the Imperial College NHS Trust. And so our experience was also kind of like bridging that gap and trying to essentially build confidence that academics actually know how to develop diagnostics. And it was kind of an exercise in knowing what the best approach around that was, in fact. And so I suppose you kind of start at the beginning, and we decided to pick a liquid handler that was very kind of programmable and we thought scalable and robust. And it was kind of a serendipitous choice because we use these liquid handlers in our own lab. Sorry. So we decided to go with the synbio Felix as our main platform that we use. And that's also because it's quite small. So in comparison to Hamilton's and TCANs, laboratories were quite receptive to this because it's such a small platform. And then we basically went about trying to design this workflow. And so there were different considerations. So it had to be resilient in terms of different reagents, different consumables, also quite easy to troubleshoot. And we tried to engage clinical scientists from the NHS as soon as possible, because it's quite easy to develop a kind of entire workflow in isolation. And then when you finally get to implementation, it's very difficult because someone goes, well, that's actually not what we need. And it was quite obvious from the beginning that what many people in industry thought were the bottlenecks were in fact not the bottlenecks. Like, still, I would say now that the bottlenecks are not around reagents. The bottlenecks are more towards the front end of the process, going from the sample tube into a 96-well plate to actually do your entire process. So again, a lot of this was it was built around building credibility, essentially, with what we were doing. And just as an example, one of the first conversations that we had with some of the other people working with diagnostics at Imperial was we were developing an RNA extraction protocol with the Felix, and they asked, oh, so have you tested it with like plasmid DNA? Because they obviously don't let you anywhere close to patient samples until you've actually tested these out on lots of different things. And we'd actually selected a completely different approach. So we'd already kind of had that in mind. And so something that I had been working on for quite a while was using bacteriophages. So the MS2 bacteriophage as a process control. And these are essentially virus-like particles. You can package them with RNA or DNA. You express them in E. coli and then purify them. And they're very, very stable in a variety of different sample matrices. So you can have blood, saliva, you can have different things. And what's really, really beneficial for us was that they're also biosafety level 1. So it meant that we could use them in our normal lab, whereas COVID was classified as generally you had to deal with it in the biosafety level 3 lab. So it was very, very difficult to work with patient samples from the beginning, and you had to kind of show them that you had something that was in fact working. And so what we did was we expressed this in an E. coli, purified it, used dynamic light scattering to show that we had a monodisperse, or relatively monodisperse particles. And then in order to quantify it, we used droplet digital PCR. And so this was then the gold standard way to quantify. We then had like a lot of all these virus-like particles that we produced that we could then test with our assay going forward as a process control. And also very, very reproducible using qPCR as well. And one of the first things that we thought was that okay, we saw a lot of these shortages in terms of reagents, so we wanted to make sure that we could use different reagents from different suppliers. So one of the first primer sets was the CDC, with the CDC primer sets. And so we tested 3 different qPCR mixes using our virus-like particles and showed that essentially, for the most part, all 3 RT-qPCR master mixes were the same. And so you could build your platform around kind of being able to pick different master mixes. And just in terms of so once we've done that, just to show you how this diagnostic platform worked, you start off with your patient samples, you then do the RNA extraction on the Felix, and then you do your normal qPCR in any kind of qPCR machine that you have. So we kind of showed that the end process works, the qPCR, but then we wanted to go back and use these particles to validate the RNA isolation. And this entire process, we think, well, I mean, in an ideal case, it's 2.5 hours from end to end, but obviously once you've done the RNA isolation and you have a plate on the qPCR, you can then start another plate on the Felix. And just to show you in general how the process flow, so you would start off, let's say you had a patient sample with 1,000 copies per microliter. You take 200 microliters of the patient sample, you put it into your sample plates, and when you extract it, if you are assuming close to 100% recovery, you would end up with 15 to 20 copies of your RNA sample in your RT-qPCR. And generally, most people consider 1 copy or close to 1 copy to be the limit of detection of RT-qPCR. So just to prove that this was quite a resilient platform, we used 2 different kits, extraction kits. The one was from Iterative, which is the company that actually makes the Felix. But then we also programmed the system to work with the Promega kits, which is actually far easier to get a hold of now. There are severe limitations in terms of how many kits you can get for the Iterative. So it was quite important to build resilience around the whole system. And this was just the first kind of the first kits that we used. So we showed that there was very low intra-run and inter-run variation. And you could detect to below 1,000 copies per milliliter, which is kind of what's clinically relevant. And since then, we've also validated other kits from other suppliers just to kind of build a really robust supply chain, because that's, in the end, that's what this whole game has been about. It's about having a very resilient supply chain with many different options of different consumables. And once we've done that, we kind of proved that we could take virus-like particles as a really good standard and develop this entire workflow with the virus-like particles. But then what happens when you go into patient samples? And what we were able to show is that it translates really, really well directly into patient samples. So on the left-hand side, you have the London Biofoundry, so LBF, and Northwest London Pathology is the collaborators that we work with at Imperial. And we showed that even though we have a completely different assay, a completely different RNA extraction protocol, the results correlate really, really well between the 2 different assays. And we also showed that between the Promega and the Iterative kits, the results also correlated really, really well. And so just to show you kind of, I know this is quite a short talk, just to show you kind of like the end process. So on the left-hand side is the workflow that we have developed. And on the right-hand side is kind of the competition. So this is one of the Roche 8800 platforms that are generally kind of the mainstay for a lot of different diagnostics applications. Both these platforms have roughly the same theoretical throughputs, and you can just see the size difference. The Roche is far more hands-off, so you kind of just put your sample tubes in and push go, whereas the Felix, there are quite a few different steps that you need to do manually, but it just shows that you can kind of develop a testing workflow from scratch that's really, 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 really reliable. And we showed that it actually had very comparable performance to the Roche systems at the NHS Trust. And then just around consumables, so just as an example, these are the Promega kits that we were using. This is the qPCR mix that we were using from Thermo Fisher. This was like 160,000 pounds worth of PCR reagents, and they just ordered £1 million worth of extraction reagents because it's 16 pounds a reaction. So just in terms of what we've done since then, so that was all the initial validation, but we've been quite busy since then. And so we now have 4 full workflows at Charing Cross Hospital, giving capacity of 4,000 a day. So this is used for patient sample testing in the NHS. and also testing all staff and care homes. And then we are in the process of building up a platform at Imperial College London, which will be doing Imperial staff and student testing as well as community testing. So as part of the track and trace scheme, with a capacity of up to 5,000 a day, and this will be fully operational from January. And these were the first, so the Felix workflow that we developed was the first custom UCAS accredited workflow for SARS-CoV-2 testing in the UK. So that just means that you could use it for patient sample testing. Whereas a lot of the other flows that were published previously have not actually been UCAS accredited. So you can use them for community testing, but you can't necessarily use them for patient testing. And we've also kind of, again, as I said, it was very much a kind of supply chain game. So it's all about making sure that you have lots of different suppliers. So we've tried at least 5 different suppliers of deep well plates and tested them to make sure that we can kind of have a very robust protocol. And in terms of what we're doing next, so we're actually working with the NHS now to look at the pooling of samples for NHS staff testing. And so this is both a technical and sample tracking challenge. So, and then we're also looking, as I mentioned, to look at kind of adding further resilience to the workflow by validating other reagent kits and plastic consumables. And I suppose this is always the thing. It's kind of like at the beginning, we thought that the bottleneck was around the qPCR step, for example. And it turned out, in fact, it's like even if you assume that it was the RNA extraction, that was never really the bottleneck. The bottleneck is still just going from your patient sample tube into some sort of format that is then automation compatible. And I mean, there are limitations in the number of tips that you can currently get. There are lots of kind of things like that, which make the process far more complicated. So I'd just like to say thank you to the London Biofoundry team, so Miles, Marta, and Marco, and then to my supervisor, Paul Fremont, and also our collaborators in the NHS, Paul Randall, Artie, and Panos. Kirsten and Andrew helped a lot with the original project. And then we've also been working quite closely with Riffin, also out of the Oakland, the East Bay area, just to try and optimize a lot of these processes and to look at the pooling process going forward. Cool.
Thank you, Michael. Does anyone have any questions?
Thank you, Michael. That was really, really cool. I obviously knew about the work that you were doing at the Foundry.
Yeah, we used one of your heads.
I know.
Thank you.
And actually, Marta and Miles are in the same building. So no, I just wanted to say, like, you've done an amazing job. It's really, really impressive what you guys done, and yeah, you can be proud. And you've been super reactive and super efficient. So well done on that. One question maybe I wanted to ask was about, you mentioned that there are still a few steps that, you compared your platform to the Rush ones, and you said Rush one, and you said, okay, the Rush platform, there's much more hands-off time, right? Can you tell us about what are the blockers for you to get to that point where you have less hands-on time?
Yeah, so I suppose the main thing at the moment is just going from this, as I say, the sample tube to a deep well plate. So currently that's done by hand. Also just because it's like, so if you use a Felix, for example, it doesn't reach into different tubes. You have different tube sizes. You have, like, you can't necessarily control a lot of the variables. So often that step is done by hand. So ideally, you also just want a liquid handler that you can just slide in your tubes, and it automatically would aliquot it into different plates, which we are currently looking at. But it was just like the availability in terms of a lot of the lead times of different liquid handlers is like 3 months, which then makes it completely impractical. There's also things like reagents you have to pre-aliquot into plates. So it's like, again, you do automate that, but it is an extra step that you then have to have these plates prepared for extraction, and then qPCR as well. So currently we use the Salma, which is like a little Felix, which just does a 96 to 96 stamp, which goes from your elution plate into your qPCR and mixes. So again, that's also, I mean, it's like you could have a completely automated workflow. Like, I think the Roche systems are basically built off TCANs anyway. And so they just have multiple things together, and then they put it into a Roche light cycler at the end. So it's like, it is possible, but it's also whether you want it to be completely like that, because I think the whole idea is to build resilience in terms of like you don't want to use diagnostic equipment. The best thing in this case is you should be using general purpose liquid handlers that can then be used for research the rest of the time and can be repurposed into test machines when you actually need them. So once you've developed all the protocols, it's like ready to go. It's just like you can just repurpose them, put them in a lab that has the biosafety requirements, and then start doing patient testing. That should be the main aim. But yeah, I mean, just as an example, in the Lighthouse labs, so for those who don't know, the main labs in the UK doing community testing are called the Lighthouse labs. A lot of that initial step was manual. So going from the patient sample tube into the 96-well plate, they just had volunteers in the hood doing that step. So they didn't even automate it.
Okay, that's pretty cool. Thank you for your answer. Second follow-up question, sorry, is about data tracking. So obviously, being able to track the samples and where they are and which stage they are, and making sure you're not making any mistake, you know, in swapping samples is super important in that context. Did you do anything in particular in order to make this really strong, this pipeline really robust?
So there's always a system that they currently use in the labs, which is mostly paper. So they literally just stick barcodes in a piece of paper, and then they double check to make sure all samples are in the right place. And diagnostic people who work in diagnostics are very, very good at this process of just going from a sample tube into a plate and making sure that they do into the right well, all those kinds of things. So that was fine. And Miles actually, so one of my colleagues wrote a piece of software just so that you could scan in all the tubes of the barcodes and then associate it directly with the qPCR results at the end. Now we're trying to work with Riffin, because with pooling, it becomes a lot more complicated because you'll start off with kind of 5 sample plates and you'll go to 1 pooled plate. And then you want to be able to deconvolute at the end all the relationships between like exactly which patient samples are in which well. And so that's a much more complicated kind of problem. And so you really want a robust solution for that. You don't want to ideally have just like code that you've written, because it is if you want to roll it out successfully, it has to be quite well thought out.
Gotcha. Thank you for your answer, and thank you for the talk.
Well, if there are no more questions, I think we reached the end. Thank you for participating in the webinar. It was really, really interesting. And yeah, we are gonna keep you updated on the development of our project since like iGEM is coming at the end as well. And yeah, thank you very much again for being here with us.
Thank you, guys. It was a pleasure.
Well, thank you.
Thanks so much.
Thanks so much.
Thank you.
Bye.